Hello, and thank you so much for joining us today. Our latest webinar is Botanical Drug Development. We're going to be talking about CMC specifically. We're not getting into a lot of clinical aspects of botanical drug development. We are an analytical testing lab. It's definitely not in our wheelhouse, but there is a lot to talk about. There's a lot of CMC challenges for botanical drug development. As she said earlier, this webinar is being recorded. If you have any questions, you can put them in the Q&A and we'll try to get to them at the end. If we don't get to them, if we run long, then we will try to answer, get back to you via email with answers to your questions. So botanical drug development, there's a guidance from the FDA, Botanical Drug Development Guidance for Industry, that was published in December of 2016. They define a botanical drug as plant materials, algae, macroscopic fungi, or any combination of the three above. It doesn't include animal parts or minerals, materials derived from botanical species that are genetically derived with the single intention of producing a single molecular entity. I always have to read that exactly. Products produced from fermentation if the objective is to produce a single molecular entity, or highly purified substances, either derived from a naturally occurring source or chemically modified. In the regulations, the Food, Drug, and Cosmetic Act states that botanical drugs are drugs if they are intended to diagnose, cure, mitigate, treat, or prevent disease. In 21 CFR Part 312, it states that botanical drugs are subject to the same IND regulations as other investigational new drugs. And in 21 CFR Part 314, it states that botanical drugs submitted as prescription drugs must follow the NDA regulations. So while botanical drugs have their own guidance, they really do follow the same drug approval process. Specifically, in the Botanical Drug Development Regulation, it states that botanical drugs are subject to cGMP requirements in parts 210 and 211. So the botanical drug really is established as a drug, because it gets a little bit confusing when we're talking about a plant. A plant can be a food, it can be a cosmetic, a dietary supplement, a medical device. But then also a drug, and then it is defined as a drug by its intended use, by its labeling, advertising, and the circumstances around its distribution. The differences between traditional drugs and botanical drugs are that traditional drugs are typically a single API. That API is well characterized, and there's some simple specifications and control strategies in comparison to botanical drugs. It's pretty prescribed, the process and the requirements to get a NDA approved as a traditional drug. Botanical drugs can have hundreds of constituents. They can have unknown actives. They will have raw material variability, and in a good way, have a substantial history of human use that can be capitalized on in the drug submission, in the preclinical or the safety studies. Something that is introduced in the botanical drug development guidance is this concept of totality of evidence. And the totality of evidence is ensuring that the therapeutic effect for the marketed drug product batches are consistent. So we want to make sure that the product, when it goes to market, is having a consistent effect on the patients. And that can be difficult when we aren't sure what the active ingredients are. So we do that with a totality of evidence, and we'll go through some examples of the 4 approved botanical drugs and what the totality of evidence was for those drugs to get approved. So it can include botanical raw material control. It can include analytical testing, orthogonal methods for different chemical classes, manufacturing controls for the drug product and the drug substance, biological assays, and then clinical consistency. And so you take all of that, you tie the botanical raw material controls, the testing, whether it's analytical chemical testing or biological assays, and you tie that to the clinical results to show that if we operate within this space, and if our product looks like this, then we should have a consistent therapeutic effect. Manufacturing of a botanical product, I'm going to be using these abbreviations. So botanical raw material is where we start, BRM, which is the plant material. Then it is extracted potentially into a botanical drug substance and then formulated into a botanical drug product, a BDP. There's a lot of requirements for botanical raw materials. One of them is that you have to retain samples of your plant, the plant part, or other botanical materials from your raw material. You have to identify your harvest location. You need to have consistent post-harvest processing procedures. You need to control your foreign matter from getting into your botanical raw material. You need to identify if there's any preservation procedures, like maybe refrigeration or freezing, to prevent change in your botanical raw material. And in this case, when we're talking about the plant material, that preservation isn't just making sure that you don't have any mold or bacterial growth in your botanical raw material. It's not a food preservation necessarily, although that would be a component of it. It's also making sure that the active materials or the active constituents in your BRM aren't changing on storage. And then having handling, transportation, and storage procedures and controls. Another thing that's required for botanical raw materials is identification. And identification of plant is a little more complicated. We can't just run it by FTIR, or, typically we get retention time or spectral match on a chromatogram for active ingredient. So in this case, we're looking at organoleptic. Does it look and sometimes smell as it should? And then macroscopic and microscopic identification. The methods that we use, there's a USP 563 for ID of botanical materials. And then you also can get a reference specimen. Sometimes you can purchase one or you develop one in-house. So the requirements for botanical raw material testing are elemental impurities, so heavy metals like the top four is the mercury, arsenic, lead, and cadmium, residual pesticides, foreign materials, microbial limits, aflatoxins, and adulterants. And these are what are specifically called out in the botanical drug development guidance. There's also a USP chapter, USP 561, Articles of Botanical Origin. And in it, there is requirements or procedures for additional tests. It does provide some guidance on sampling, tests for foreign organic matter, ash, total ash, acid insoluble ash, and water soluble ash, and then alcohol water soluble extractives, crude fiber, starch, volatile oil determination, water content, aflatoxins, pesticide residue, and elemental impurities. The elemental impurities refers to the general chapter for elemental impurities, but pesticide residue actually lists all of the pesticides that you need to test. And this really is the list of pesticides that should be tested for any botanical drug is in this USP chapter. If you're looking to market your product, your botanical drug product in a different region like Europe, the EP monographs or the EP, the European Pharmacopoeia, also has a list of pesticides, and it's generally harmonized with the USP, but there's a couple of exceptions. So you want to make sure that you've looked that over and you've addressed that before you move into the European market if you've developed it for the US market. And then polycyclic aromatic hydrocarbons is a new test that's being added on the revision that becomes effective in December of this year. There's a requirement for qualitative description of your botanical drug substance, and we're kind of moving into the IND phase now. So as you're developing a product, you need to have a name for it. You need to describe it. What does it look like? Provide physicochemical properties of it, any biological activity, and then prior clinical use of each of the raw materials that are going in there. One thing that you also need to provide is how you're preparing your drug substance if there's more than one plant. So if you have a drug substance with two different plants, let's say they're at 50-50 ratio in the drug substance, are you mixing the plant material together at the same time or at the beginning before you extract? Or are you doing a extraction and then mixing that botanical drug substance together at that point? So you need to provide information on how you are doing that manufacturing. And then you need to give a quantitative description. So the strength as a dry weight of your processed botanical drug substance, batch size and yield relative to your BRM. So that would be if I extracted a kilo of BRM, how much botanical drug substance am I getting from that extraction? And the amount of the active constituents, if known. And we talked about that a little bit earlier. There are some botanical drugs where it would be difficult to identify the active constituents in it. And so if you know the active constituents and you know how much are in there, you need to provide that information. And this, I think there would be an expectation based on available literature information. If there's information out there on what the active constituents of your botanical drug substance are, or the botanical raw material, the expectation would be that you are characterizing that according to the available information. And then the composition of your multi-plant BDS as a relative ratio of the botanical drug substance or the botanical raw material, depending on what your manufacturing process was. So how much of each of the plants are you mixing together, whether that's BDS or BRM? So quality control for the botanical drug substance, we're looking at appearance, we kind of redundant to the last slide, but strength by weight, identification of your active constituents, and typically chemical identification is done by HPLC with a retention time and UV match, maybe LCMS, and then quantification of the active constituents, again, if known, or a biological assay. And the biological assay isn't something that is required for every botanical drug, but it is a tool that can be used if it's difficult to chemically characterize the botanical drug substance. And then mass balance of your BDS. And mass balance is looking at things like lipids, carbohydrates. We'll go to a hemp example because that's the space that we work in a lot. If you have a hemp extract, we know that one of the primary constituents is CBD. There's other cannabinoids in there. We also know that there's terpenes in there, potentially some water, depending on your BDS. And so looking at adding all of that together to see how close you get to 100. So mass balance is how close to 100 are you? How well can you characterize your BDS to understand everything that's in it? And then residual pesticides. Your botanical drug product, a lot of the characterization has happened in the drug substance. So in the drug product, your qualitative description is the dosage form, your route of administration, and the name and functions of all of your ingredients. And the quantitative really is the strength. And in this case, or in some cases, it would be the chemical constituents. In other cases, the strength would be characterized by your biological assay. We're moving now into phase 3 of your IND, later development. So it is expected that you're going to have batch-to-batch variability of your botanical drug substance, but you need to establish what the impact of that variability is on the therapeutic effect. The best way to do that is to use different lots of your botanical raw material in your clinical studies, in your phase 3 studies. And in the guidance, it recommends that you're looking at 3 different growing regions for use in your phase 3 clinical study. You want to compare your IND batches that you've manufactured to any batches that you've referenced. So that would be preclinical studies. So any toxicology studies that you are referencing from literature, you need to be able to understand the chemical, hopefully, the chemical composition of those. Is a lot easier if it's chemical, or the biological assay. So you need to be able to draw a line between the product that you're manufacturing and any of the batches, any of the data, any of the clinical or preclinical data that you're referencing. You need to detail any manufacturing process changes throughout your development. And then you have to retain, or you really should retain, sufficient material for further characterization studies. So this is keeping some of your BRM, keeping some of your BDS and your BDP, so that as the development continues, you're able, and you've identified an active constituent in your BDS, you can go back to product that was used in your clinical study and analyze that for that active constituent, and then be able to draw a line back to the clinical data that you generated previously. It could be that as you're going through the review process with the FDA, they ask you to characterize a certain chemical entity. And then this way you have the materials to be able to go back and test it once that methodology is established. So the BRM chemistry manufacturing controls to support phase 3, again, establish large growing regions with 3 or more cultivation sites or farms. You want to follow the principles of GACP, which is good agriculture and cultivation practices. And you should do some additional work on your BRM, or you should have done already some additional work on your BRM characterization. So chemical ID, and then potentially some authentication of the BRM by DNA fingerprinting. And that really is, if you have tight controls into what cultivar or what species you're using, you might need to provide additional information. And then any updated analytical methods and your proposed specification acceptance criteria. So the expectation isn't, as you go into your phase 1, phase 2, that you really have fully characterized your drug substance. And that's work that needs to continue on. And so you wouldn't have finalized specifications early in your IND phase. You would want to have those evolve as you manufacture more BDS, as you manufacture, as you have more BRM available to process. For the BDS and the BDP, for phase 3, you provide updates on the additional characterization work that you've done. You have your specifications with your interim acceptance criteria that you're going to finalize during your NDA review. Your pharmaceutical development of the BDS and the BDP should be well advanced, and because you really don't want to have any significant changes in your manufacturing process once you get to phase 3. Phase 3, really everything should be locked in, and you are taking to clinic exactly what you are going to be taking to market. So moving on to the NDA, the new drug application, the submission requirements are generally the same as those for other drug products. There's one exception, we'll talk about that in a little bit. The pre-IND meeting, pre-NDA meeting is recommended for you to meet with the FDA and agree on the content of a complete application. I think FDA wants to hit their PDUFA goals, And to do that, they want to make sure that when you file your application, you know exactly what they need for you to put in the application for it to be able to be approved. And then the submission, as with everything else, is in eCTD format. So in the NDA, you want to have clinically relevant specifications, and we've talked about this a little bit already. You need to be able to tie your specifications, whether that's for chemical assays or biological assays, back to clinical studies where the therapeutic effect was demonstrated. You're going to need to have orthogonal analytical methods. You'll have methods for different, if you can characterize it, different classes of compounds. We'll go back to the hemp example, cannabinoids and terpenes. You want to provide data to address mass balance, which hopefully you can achieve, and in-process controls and testing established for your BDS and your BDP manufacturing. So, when you can't fully characterize your BDS, when you don't really know what all of the active chemical compounds are, it is possible to establish your ID based on a fingerprint of the chemical constituents. And there's an example of that in one of the INDs that has already been approved. So a quick note on biological assays. They measure the activity, potency, or effect of a substance using a biological system, such as cells, tissues, microorganisms, or enzymes. So when chemical testing alone is not sufficient to ensure the quality of the product and the therapeutic consistency, you can use a biological assay. It should reflect the drug's mechanism of action. And if you have multiple indications, it might be required that you have multiple biological assays. And each of those biological assays have to be validated. And I think the general chapter for biological assays is 1032. So there's a whole USP, it's quite extensive, on biological assays, development, and validation. System suitability controls and things like that as well. Stability studies are required for botanical drugs. And in this case, I think that the ICHQ1A is the only ICH that is referenced in the botanical drug guidance. And in all of the ICH guidances, as we know, I really like my ICH guidances, they specifically say that herbal drugs are outside the scope of those guidances, with the exception of Q2, which is method development and validation. So the specifications, impurities, I think the specifications, impurities, even elemental impurities, say that herbal drugs are not applicable. So stability studies, again, required. Your methods need to be stability indicating. We haven't talked a lot about the analytical challenges with developing methods for botanical drugs. I don't have a lot of data that I don't have any data that's outside confidentiality that I can share. So, the methods that we have developed for different customers look at upwards of 100 or 200 different peaks in those chromatograms. So developing stability indicating methods is really challenging. Trying to find a method where you have degradants that are showing up that aren't interfering with another constituent. A lot of times, though, the degradants that we see, for us, luckily so far, are already known compounds. So that helps a lot. You should include biological assays as applicable if you have one. And then you should perform stress testing to identify degradation products in the BDS and the BDP. And again, stress testing then isn't required, or I don't think it's required for biological assays, because you're not really looking at degradation products there, right? You're just looking at biological activity. And then you need to monitor any degradation products throughout the stability. We use that stability data to establish the retest period for the botanical drug substance and the expiration dating period for the botanical drug product. Specifications for your botanical drug product and your drug substance need to be clinically relevant. So they need to be tied back to the product that was used in the clinical study. Your analytical procedures should all be validated, and they should be sensitive enough to detect any differences in the critical quality attributes you would determine during the development process. When you're using orthogonal methods, so again, we're talking in the hemp example, uh, cannabinoid and terpene testing. Um, you need to take the data and look at it together to try to demonstrate the mass balance. So total amount of terpenes, total amount of cannabinoids, whether they're known or unknown for both of them. But then you should also report individual constituents within the different chemical classes. If a biological assay is needed for quality control, you should evaluate mass balance in conjunction with the biological assay. They're in the botanical drug development guidance. They are very specific. This is me writing this out exactly what they're asking for your drug substance specification. They don't do this for the drug product. So they want to know the amount by weight of each known chemical constituent. They want to know the total amount of the constituents in the same class. So again, individual cannabinoids and total cannabinoids. They want to know the area percentage for each known peak, and they want the relative retention time for each unknown quantifiable peak. So anything that is above the LOQ of the method, they want to know the relative retention time of those. They want to know the amounts by weight of the total lipid in individual fatty acids, and the amounts by weight of total amino acids and individual amino acids. And the same for the simple and complex carbohydrates. And then the amount by weight of total and individual vitamins. And then the last one is ash content. And if you don't know what ash is the inorganic material that is left over after all of the organic matter is burned off. So we use a muffle furnace and we put it at, I think, about 600 degrees and burn all of the organic material off and look and see how much ash is left. The manufacturing controls for your NDA submission, your control of the BRM should be demonstrated. You should have controls for BRM manufacturing, and you should present those in your NDA. You should not rely on the BDS and the BDP controls. So that means you should have control over the botanical raw material that is being grown. You should have procedures in place for storage, handling, and processing of the BRM. The BRM controls may include both GACP, which is good agricultural and cultivation practices, and CGMP, depending on how the material is grown, collected, processed, and stored. And it kind of depends also on where you are initiating your drug substance manufacturing. So if your drug substance manufacturing starts at the cultivation site, then GMP would definitely apply. And then the beginning of your BDS manufacture should be designated. So where do you start your BDS manufacturing? Is it with milling of the BRM? Is it with the drying? Or is it with the extraction? You need to designate that and then apply appropriate GMP controls at that stage. I said that there was one slight difference in the NDA for botanical drugs, and it's this section, assurance of therapeutic consistency. I think we could also call this totality of evidence, right? So This is a unique section for botanical drugs. It's in module 2.3.p.2, pharmaceutical development, and it should include a summary and an integrated evaluation of raw material controls, quality control, chemical tests, and manufacturing control, biological assays, and your clinical data. So it should be a discussion on how all of these different parts of your control strategy are coming together to ensure that the product you manufacture during your clinical trials is going to be the same product that you're manufacturing after approval, so that you are assuring, this is how we're assuring that the therapeutic effect is going to be the same as it was during the clinical studies when we start selling it. This right here, this assurance of therapeutic consistency, is the biggest, I think, challenge, and I want to say the biggest gift that the FDA has given us, because it would be nearly impossible to get a botanical drug through the typical NDA approval process with just chemical assays, depending on what it is and depending on how processed it is. But this right here, this section is a section that you need to add to your NDA to explain your control strategy for your botanical drug. So the current botanical drug landscape, as I said, there's 4 FDA-approved botanical drugs, and several, I think over 100, I can't remember the number, botanical INDs that are in process. So the therapeutic areas for the INDs are listed there: oncology, neurology, dermatology, psychological, and cardiovascular. So here are the botanical drugs. Again, 4 of them, 3 of them are topicals and one's an oral. And then 3 of them are NDAs and one's a biologic. You can see the different botanicals that are used. There's a really great review article on botanicaldrugs.org. I think that I have probably gone back to this article a dozen times since I found it I think I found it in March. I found it really early. But it is really helpful. I think it is such a great assessment of the CMC that was used to gain approval of these 4 NDAs that have gotten approval from FDA. So here's a list of the drugs, and then the totality of evidence, and this is directly from that article. I recommend that you read it. If you're doing botanical drug development, you should probably have already read this. If you haven't, get there and read it. There's a lot of really good information, a lot of really good clinical information that we didn't go into at all here. So the totality of evidence that is used for the first drug there, Verigen. Sorry, I'm horrible at pronouncing these. Raw materials are restricted to cultivars used in clinical studies. So they put tight controls around the raw materials. The chemical specifications were established from levels in the clinical trials, and the clinical data indicated that the compositional variability of the drug product was not significant. So they had drug product that went into their phase 3 study that had high variability, and the clinical data indicated that that wasn't significant. And that right there allows you to, it gives you a lot of rein on your specifications, and also in maybe where you're growing your product or growing your BRM, it gives you some leeway there. Mytesi, it's the totality of evidence for that one. Harvesting restricted to eco-geographical regions, so specific regions where they can grow. They had a clinically relevant biological assay, and then they demonstrated dose insensitivity during their clinical study. So they had, I don't remember the numbers, but I think it was almost like 100 milligrams and 200 milligrams that went into their study and demonstrated that the response wasn't very sensitive to the dose. And so that allowed some variability in the manufacturing of it. The next one, they did a quantitative triterpene profiling and then had a broader triterpene fingerprint that they used for ID. They had processing controls for the ologel extraction and formulation, and then correlated again, same thing, correlated batch variability to clinical efficacy. So as you're going through your drug development, it's really important to have a good regulatory strategy for your manufacturing to make sure that you are appropriately looking at a variety or variable drug substances so that when you go into your manufacture of your commercial product, your specifications and your controls aren't so tight that if something changes, you can't manufacture. So it's a regulatory strategy. You need to look at the risk because there's risk of going into a clinical study with product that has some variability and demonstrating that it's not efficacious. But that's good information to have. It's good information to have. But hopefully you can show that with your botanical drug, batch variability doesn't have a huge impact to your therapeutic efficacy. And then the last one, again, it was characterization by functional activity, which is a biological assay rather than chemical composition. And then they had some controls on their extraction and purification process. This last one was the biologic, so that's a little bit different of a pathway. So the key takeaways that I want you to have from this presentation is that botanical drugs really require a different CMC mindset. This totality of evidence, the idea that you're not just setting a specification, that you're not just looking at chemical composition, that you're looking at maybe biological activity and you're putting in process controls, you've got your BRM controls, right? So the quality comes from controlling the entire system, not that single API. Your analytical characterization should evolve throughout development if you are characterizing the chemical composition. It should evolve throughout development. You should be working on further identifying, characterizing all of the different chemical constituents as you are developing a product. And then early CMC planning will help reduce regulatory risk and risk to not being able to manufacture your commercial product and should accelerate development. So thank you so much for joining us today. I really appreciate it. I hope that you got something good out of this. So I did have one question. And so how easy is it to set specifications for botanical drugs? What should I take into account? That is a really good question. I would say how easy, not easy, depending. So if you have a more purified botanical drug substance, it would be easier. If you had tight controls over your BRM, it would be easier to set those specifications. But the process that you should go through for this is to, as you're starting your development process, characterize your BRM, characterize your drug substance. If you've been to our previous webinars and we talked about analytical development and specification setting, I talk about how important it is that the analytical methodology that you're using in early development is either the same, so you've done a lot of work to develop your analytical method really early, or you can bridge the analytical method to a new analytical method later. Because when you analyze your early batches, we want to take those batches along with every batch you manufacture all the way up to and through phase 3 and probably into any kind of validation batches. You want to put all of those together you want to look at the variability from BRM lot to BRM. You want to look at then the subsequent variability that you're seeing in your BDS. And when it comes to your botanical drug substance or your botanical drug product, really at that point, you should just be looking at degradants and changes. You don't have to look at the impurities and all of that. But so you want to take all of that batch data and make a massive table, then take into account changes that you see on stability or any stress testing. And from there, then you make an assessment of this is what my specification should be. And I think that you'll find as you go through the process and you go from harvest to harvest and BRM lot to BRM lot and subsequent BDS manufacturing lots, you'll start to see places where there is significant variability and places where there isn't very much variability. And so you take that data and use that to write your justification of specification, either report or the section. And I always did a justification of specification report where I went analyte by analyte and justified based on the data what the specification should be. So the other thing that you want to take into account, obviously this is a hot topic for me, the other thing that you want to take into account is analytical variability. If we are talking about a low-level analyte, so an analyte that maybe is less than 1%, then your analytical variability increases significantly once you start looking at those lower concentrations. So if we're looking at something where the specification is around 1%, we need to take into account that there's going to be some analytical variability as well as BRM, right? As well as the variability coming from the cultivation, and then potentially some variability from your process. So all of that data you generate, and the more data you can generate, the more batches you can make before you get to that phase 3, and certainly before you get to your NDA, the more robust your specifications are going to be, and the more likely your specifications are going to allow for consistent manufacturing and consistent release after approval. The other thing, like talking about specifications, I haven't done a biological assay, so I can't really speak to the biological assay specifications, but the way they're put together is as a comparison to a standard. So you would evaluate the activity of your drug substance or your drug product, and then compare that to a known standard that has known biological activity, and it would be a percentage of that. So hopefully that answers your questions on specifications. I think that, pretty much I was addressing chemical and not necessarily pesticides, heavy metals, residual solvents, and all the other things that need to go on there, but those are pretty well characterized. And even though the ICH states that those guidances aren't relevant to botanical drugs, I think that there's still really good guidance on what your limits should be and in some cases methodology. So excellent. And I think that's it for questions. Again, thank you so much. If you have any other questions, feel free to email us at engage@triveritylabs.com, and we will see you on the next one.